Hello there. Welcome back to Midnight Ocean. I'm Velen, your guide to traveling through the wonders of the ocean. Tonight, we're going to explore the mesopelagic zone, also called the Twilight Zone, the second level of the ocean's depths. If these cozy moments calm you, I invite you to subscribe, share a comment, or leave a like. Now, get comfortable, take a deep breath. Don't be afraid, you're safe here. Let's dive deep into the sea and let our imagination guide us to a peaceful and deep night. Around 1942, United States Navy ships equipped with the newly invented sonar made an intriguing discovery. Operators were puzzled by an unexpected acoustic signature they called the phantom bottom. It looked like a solid plane about 300 to 500 meters below the ships. The actual depth of the ocean at that point was thousands of meters. Multiple explanations were proposed, including equipment failure or some enemy sonar jamming device, but none held up under investigation. In 1945, Martin Johnson of the Scripps Institution of Oceanography proposed a solution. This deep scattering layer was made up of living creatures, trillions of shrimp, jellyfish, and bony fish that rose every night to feed in warmer surface waters before returning to their deeper homes. That false bottom was in fact life, concentrated in a specific band of the ocean now called the mesopelagic zone. Operators trained to trust their instruments came up against an impossible floor, present at night and absent during the day. The mystery persisted for years without a convincing explanation. Technicians suspected defects in the newly built transducers, recalibrating equipment over and over. Only by comparing records between different vessels, in different oceans, did a pattern emerge that was too consistent to be mechanical failure. The solution required cooperation between the military navy and civilian scientists, uniting wartime strategic necessity with biological curiosity. The episode shows how new technology often reveals phenomena researchers never imagined existed, hidden beneath a surface everyone assumed they already knew. Sonar works by emitting sound pulses and measuring the return time of the echo reflected off solid surfaces. Submarines, submerged rock formations, dense schools of fish. Any sufficiently compact object reflects sound waves in a detectable way. Operators interpreted consistent echoes as evidence of fixed terrain, since marine biology rarely formed layers extensive and uniform enough to simulate a genuine ocean floor. No one anticipated that small organisms, aggregated in immense densities, would reflect sound with an intensity comparable to rock. The discovery exposed a limitation of the technology. Instruments record raw data, but correct interpretation depends on prior knowledge about what's being measured, knowledge that didn't yet exist. Swim bladders, gas-filled internal organs present in many fish, explain part of this intense acoustic reflection. The compressed gas creates a sharp density difference relative to the surrounding water, reflecting sound waves efficiently. When billions of small fish, each carrying this gas-filled bladder, concentrate in a relatively thin layer of the ocean, the collective effect approaches a continuous surface for acoustic purposes. Scientists later measured this reflection, relating signal intensity to estimated population density. The technique that began as a military mystery became a valuable tool for monitoring mesopelagic fish populations without physically capturing every specimen. Martin Johnson was part of a growing scientific community interested in ocean ecology in the post-war period. Submarines, sonars, and other military technologies found peaceful applications in scientific research soon after. Scripps became an important hub for investigating this mysterious layer. Subsequent expeditions collected samples with specialized nets, capturing specimens that confirmed the initial hypothesis. The analysis revealed wide diversity: lanternfish, hatchetfish, various shrimp, small squid. Each species contributing to the collective acoustic signature that had confused sonar operators years before. Recognition of this biological layer changed our understanding of the ocean's vertical structure. Before, it was assumed that marine life was distributed relatively evenly throughout the water column, with density simply decreasing as depth increased. The discovery revealed a more complex organization. Distinct zones, each with its own physical characteristics, hosting communities specifically adapted to those conditions. This stratification became a central concept in modern biological oceanography, shaping how later research is planned, always considering depth as a critical variable for determining which organisms can be found at a given location. The military implications also drew attention at the time. Submarines could, in theory, use this biological layer to mask their own acoustic signature, confusing enemy detection amid the noise generated by trillions of organisms. Naval strategists began treating this layer as a relevant tactical variable, not just a scientific curiosity. At the same time, the discovery underscored just how unexplored the ocean remained, a massive biological structure, even affecting military operations, had gone unnoticed for decades of navigation. The episode became a reference point for how tools built for one specific purpose can reveal entire dimensions of the natural world. Decades later, the deep scattering layer continues to be studied with technologies far more sophisticated than the original sonar. Modern echo sounders map this structure with impressive precision, revealing seasonal, regional, and even daily variations in its thickness and density. Satellites and acoustic sensors distributed throughout the oceans allow for continuous monitoring, something unthinkable for the operators of 1942. This technological legacy directly connects the wartime urgency of a world war to today's scientific understanding of deep marine ecosystems. The phantom bottom that puzzled sailors has become one of the most consistently studied subjects in contemporary biological oceanography. The story of this acoustic illusion stands as a reminder of how much human knowledge about the ocean still depended, and still depends, on lucky accidents during investigations carried out for other purposes. No scientist set out to discover the planet's largest biological layer using military equipment meant to detect enemy submarines. The initial confusion, far from being a failure, became a gateway into an extensive chapter of marine biology, one that's still expanding as new sensing technologies reveal additional details about this in-between band of the ocean, where sunlight fades and a massive migration of organisms redefines, every single night, the boundaries between surface and depth. The mesopelagic zone extends from about 200 to 1,000 meters in depth, and is sometimes called the Twilight Zone, because sunlight can still be detected here at low levels. The light that reaches this band isn't enough to sustain photosynthesis, but it is enough for adapted eyes to still detect shadows, silhouettes, and movement. Temperature drops significantly, from over 20 degrees Celsius at the upper boundary to about 4 degrees Celsius at the deepest extent. Pressure increases by roughly 1 atmosphere for every 10 meters of depth. This zone corresponds to about 60% of the planet's surface and roughly 20% of the ocean's total volume, making it a significant part of Earth's biosphere. By volume, it's one of the largest continuous environments on the planet, larger than all the world's tropical rainforests combined. Picture slowly diving down from the sunlit surface, watching colors progressively disappear as the water absorbs different wavelengths. Reds vanish first, within the very first few meters, then oranges and yellows fade gradually. By the time you reach 200 meters, a deep, faint blue dominates, insufficient for any plant to carry out photosynthesis. This transition marks the entry into the mesopelagic zone, where organisms rely on sophisticated sensory adaptations to survive in constant dimness. There's no total darkness yet, but there's too little light to guide behaviors based on conventional vision, demanding creative biological solutions for finding food, avoiding predators, and finding mates in this in-between band. Hydrostatic pressure increases steadily and predictably as depth grows. Every additional 10 meters of water adds roughly 1 atmosphere of pressure, equivalent to the weight of the entire Earth's atmospheric column pressing down on the surface. At 1,000 meters, organisms face about 100 atmospheres, pressure capable of instantly crushing structures not built for it. Mesopelagic animals have evolved bodies without compressible air cavities, avoiding the collapse that would affect rigid structures. Many have gelatinous tissues or internal fluids whose pressure naturally balances with the surrounding environment, allowing normal function in conditions that would destroy any land-based structure almost instantly. The drop in temperature accompanies the increase in depth just as sharply. Surface waters, warmed directly by solar radiation, can exceed 20 degrees Celsius in tropical regions. As depth increases, that thermal energy progressively dissipates, resulting in temperatures near 4 degrees at the lower limits of the mesopelagic zone. This thermal difference creates stable vertical stratification, making it harder for layers to mix. Organisms that migrate daily between warm surface zones and cold depths face considerable metabolic challenges, requiring specific physiological adaptations to tolerate significant temperature swings during regular vertical movements. The scale of this zone is surprising when compared to familiar land environments. Covering roughly 60% of the planet's surface, the mesopelagic band exceeds the size of any known continental biome. Tropical rainforests, often cited as the primary repositories of terrestrial biodiversity, cover a much smaller area by comparison with this intermediate ocean layer. 20% of the total volume of the oceans corresponds to this twilight zone, a number that, translated into cubic kilometers, exceeds any known geological structure on land. This scale underscores just how much remains unexplored compared to land environments that have already been extensively mapped and cataloged. Visual adaptations among the inhabitants of this zone reveal remarkable evolutionary solutions for capturing scarce light. Many mesopelagic fish have disproportionately large eyes, maximizing the area available for capturing remaining photons. Specialized retinas concentrate light-sensitive cells, optimizing sensitivity even under minimal lighting. Some organisms have developed tubular, upward-facing eyes, allowing them to detect the silhouettes of predators or prey against the faint residual glow coming from the distant surface. This ocular specialization illustrates how specific environmental pressures shape precise biological structures, adapted exactly to the particular light conditions of this in-between band. Bioluminescence becomes a widespread strategy in this permanent dimness. It's estimated that most mesopelagic organisms produce their own light through internal chemical reactions, using this ability for communication, camouflage, or hunting. Counterillumination, a technique where animals emit light on the underside of their body, matching the faint light coming from above, makes silhouettes less visible to predators looking up from below. Others use light flashes to confuse attackers or lure curious prey. This widespread production of biological light turns the mesopelagic zone into a visually active environment, even in the near-total absence of external light from the sun. The chemical makeup of the water also varies with increasing depth in this in-between band. Dissolved oxygen concentration progressively decreases, creating regions known as oxygen minimum zones in certain ocean locations. Organisms adapted to these conditions have developed efficient respiratory systems, capable of extracting oxygen even from comparatively oxygen-poor waters. Reduced partial pressure of dissolved gases requires specific metabolic adaptations, including differentiated respiratory rates and specialized blood pigments to maximize the capture and transport of available oxygen under these challenging conditions. Researchers classify this zone as a crucial ecological boundary, separating light-rich environments from the completely dark depths below. It functions as a transition zone where survival strategies adapted to sunlight gradually give way to strategies based on other senses, such as detecting vibrations, dissolved chemicals in the water, and bioluminescence. Understanding this transition helps scientists map how life organizes itself vertically in the oceans, revealing patterns applicable to practically every ocean basin on the planet, regardless of latitude or the specific geographic features of each region studied. Recognizing the vastness of this twilight zone changes our perception of where, exactly, most of the planet's animal life actually lives. Far from the popular images of colorful reefs or busy coastlines, the largest volumetric share of animal biomass likely resides in this in-between band, dimly lit and rarely observed directly by humans. This reality reshapes scientific priorities, directing growing attention toward a region that, despite its monumental scale, remains considerably less studied than surface or coastal environments, still hiding fundamental ecological processes essential to the functioning of the entire ocean system. The phenomenon that confused the Navy's sonar in 1942 has a technical name: Deal vertical migration. Billions of animals, including fish, zooplankton, and squid, rise from the mesopelagic zone to food-rich surface waters at night. They feed under the cover of darkness, then descend back into the deeper, darker mesopelagic zone before dawn. This synchronized migration is primarily a strategy to avoid visual predators that hunt in the sunlit upper layers during the day. Many organisms in the mesopelagic zone move into the epipelagic zone at night and return to the mesopelagic zone during the day, a pattern known as Deal vertical migration. This movement represents the largest synchronized biomass migration on Earth, influencing ocean food webs and carbon cycles. No land migration, not the wildebeest migration across the African savanna, nor the caribou migration in the Arctic, comes anywhere close to this scale. Picture a biological clock synchronized among trillions of distinct organisms, going off simultaneously at dusk in practically every ocean on the planet. This coordinated movement doesn't depend on direct communication between species. It results from individual responses to shared light cues. As sunlight intensity drops at dusk, photosensitive receptors in millions of different organisms trigger similar signals, kicking off a nearly simultaneous collective ascent. This pattern has repeated itself daily for millions of years, making it one of the most consistent and predictable biological rhythms on the planet, comparable in regularity to the very day-night cycle that triggers it in the first place. The advantages of this migratory strategy explain why it persists across such a wide range of species. Surface waters concentrate phytoplankton, the base of the marine food chain, which thrives wherever sunlight allows for efficient photosynthesis. Staying permanently in these nutrient-rich waters, however, would expose small organisms to visual predators, larger fish, seabirds, marine mammals, that hunt efficiently under abundant light. Migrating vertically allows organisms to exploit surface food resources during nighttime darkness, when visual predators lose their effectiveness, then return to the depths where mesopelagic dimness offers additional protection from visual detection during the more dangerous daylight hours. The scale of this migration defies comparison with any known land-based movement. It's estimated that the total biomass involved exceeds billions of tons, moving vertically hundreds of meters twice a day, every day, across practically every ocean basin simultaneously. Famous land migrations, like the seasonal movement of large African mammals, involve substantial but geographically limited populations, occurring only during certain times of year. The mesopelagic migration happens globally, daily, and involves a vastly greater taxonomic diversity, including fish, crustaceans, mollusks, and various gelatinous organisms, making it a truly planetary phenomenon in both scale and frequency. The speed of this vertical movement varies by organism, but many species can travel hundreds of meters within a matter of hours. Small crustaceans, propelled by efficient swimming appendages, continuously adjust their position in the water column as light levels change throughout the day. Larger fish, with more developed muscles, move even faster, optimizing the time spent in transit to maximize the period available for both surface feeding and deep refuge. This constant rhythm of rising and descending represents considerable energy expenditure, presumably offset by the survival benefits the strategy provides across successive generations. The ecological consequences of this migration extend well beyond the organisms directly involved. Larger predators, including certain species of whales, tuna, and sharks, adjust their own hunting patterns to coincide with the predictable presence of these migrating masses at different depths throughout the day. An entire food chain organizes itself around this regular vertical rhythm, creating complex temporal dependencies between species that would otherwise rarely cross paths. This ecological synchronicity illustrates how a single behavior, replicated on a massive scale, can structure feeding relationships across multiple trophic levels at once. The role this migration plays in the global carbon cycle is drawing increasing scientific attention. Organisms that feed on phytoplankton in surface waters at night carry carbon stored in their tissues as they descend back to mesopelagic depths. Metabolic waste, fecal particles, and sinking carcasses actively transport carbon from the surface to deeper layers, a process known as the biological carbon pump. This vertical transport mechanism, driven directly by the daily migration, contributes significantly to atmospheric carbon sequestration in the oceans, making this animal behavior relevant even to global climate models. The scientific methods for studying this phenomenon have evolved considerably since the first confusing sonar readings back in the 1940s. Modern echo sounders, permanently installed at certain ocean locations, continuously record the depth of this biological layer, documenting daily, seasonal, and even moon phase-related variations. Specialized trawl nets, operating at different depths and times, allow researchers to capture representative samples for species identification. More recent acoustic technologies can even distinguish between different taxonomic groups based on specific characteristics of their reflected echoes, substantially refining our understanding of the composition of this massive migration. Variations in this migratory pattern occur as specific environmental conditions change across different ocean regions. In polar waters, where periods of light and darkness vary dramatically throughout the year, the migratory pattern adjusts accordingly, sometimes temporarily disappearing altogether during periods of continuous light or darkness characteristic of high latitudes. Moonlight also influences migration intensity. Brighter nights can reduce the magnitude of the ascent, since visual predators retain some effectiveness even under moonlight. These regional and seasonal variations demonstrate the phenomenon's sensitivity to precise light conditions, reinforcing light as the primary driver of this behavior. Understanding this massive, daily migration reshapes perceptions of ocean dynamics that many consider static or slow compared to land-based processes. Every day, an amount of biomass equivalent to entire populations of large land mammals moves vertically through hundreds of meters of water, repeating this cycle without interruption for ages. This movement remains largely invisible to human observers on the surface, except through specialized instruments like the ones that confused sonar operators decades ago. The same layer that once looked like a false ocean floor turns out, in the end, to be a biological system in constant motion, connecting sunlight, food chains, and planetary chemical cycles in a daily choreography that continues night after night in practically every ocean on Earth. Recent studies suggest that fish biomass in the Twilight zone may be 10 times greater than previously thought, more than in the entire rest of the ocean combined. Although typically small, mesopelagic fish make up the largest fish biomass in the world because of their sheer numbers. Earlier estimates put mesopelagic fish worldwide at around 1,000 million tons. Using data collected during the Malaspina 2010 circumnavigation expedition, researchers showed that there are roughly 10 times more mesopelagic fish than previously estimated, somewhere between 10,000 and 15,000 million tons. To put that in perspective, that estimated mass of fish is equivalent to dozens of times the entire human race's annual fishing catch combined, living in a single band of the ocean that most people have never even heard of. Picture gathering up an entire year's worth of the world's commercial fishing, tuna, sardines, cod, salmon, and realizing that amount represents just a small fraction of what's swimming unnoticed in the mesopelagic zone. Earlier estimates already seemed impressive. The later revision multiplied that number by 10, revealing just how much science had underestimated this environment. This kind of drastic correction isn't common in biology. Normally, refinements adjust estimates by modest margins. A tenfold multiplication signals how little was known about this region before advanced acoustic technologies and dedicated expeditions made a more accurate count of this colossal biomass possible, hidden beneath hundreds of meters of dark water. The Malaspina expedition, named after a historic navigator, traveled the world's oceans between 2010 and 2011, collecting oceanographic and biological data on an unprecedented scale. Scientists on board used echo sounders specifically calibrated to detect the deep scattering layer across different latitudes and longitudes, supplementing acoustic records with physical catches for calibration. This combination of methods allowed for a significant correction of earlier models, which had relied mainly on traditional fishing nets, instruments that aren't very effective at catching small, agile mesopelagic fish, which can dodge these nets with considerable ease during collection operations. The discrepancy between old and new estimates stems partly from technical limitations of conventional nets. Mesopelagic fish, many equipped with sophisticated sensory systems for detecting predators in dim light, often sense the approach of trawl nets and escape before being caught. This systematic evasion led to severe undersampling in historical surveys, resulting in artificially low population estimates. Acoustic methods, since they don't rely on direct physical capture, get around this limitation, registering biological presence regardless of the organism's own ability to evade capture, offering a more accurate picture of the actual abundance present in this in-between layer. The magnitude of this revised biomass directly implies recalculations for ocean carbon cycle models. Organisms that feed at the surface and descend to mesopelagic depths carry carbon incorporated into their bodies, contributing to the sequestration of that element away from the atmosphere. If the real population is 10 times larger than earlier estimates suggested, the volume of carbon transported by this daily migration also needs to be recalculated proportionally. This adjustment directly affects climate models that depend on accurate estimates of how much carbon the oceans absorb and store, making this biological correction relevant to global climate science. Commercial interest in this massive biomass is already drawing attention from the international fishing industry. Some countries are exploring the possibility of targeted fishing of mesopelagic fish for fish meal, oil, or dietary supplement production, given the apparently available population scale. However, scientists warn of significant risks associated with this potential exploitation. The ecological role this biomass plays in the carbon cycle and ocean food chains remains insufficiently understood to guarantee sustainable extraction without unwanted consequences. Removing substantial amounts of this population could destabilize ecological processes whose importance the scientific community is only now beginning to fully recognize. The geographic distribution of this biomass isn't uniform. Concentrations vary depending on regional surface productivity, water temperature, and nutrient availability. Ocean regions with upwelling of nutrient-rich deep water tend to support denser mesopelagic populations, since abundant phytoplankton in these areas feeds the entire subsequent food chain. Mapping this distribution, made possible by modern acoustic technologies, reveals patterns correlated with known ocean currents, giving researchers additional tools to predict where particularly dense concentrations of this biomass are likely to form at different times of year. Comparisons with land-based biomass help put this discovery into context for general audiences. The world's entire cattle biomass, often cited as a benchmark because of its impressive scale, comes in at around a few 100 million tons. The revised mesopelagic biomass exceeds that number several times over, even though it's made up entirely of individually small organisms, some measuring just a few centimeters. This comparison highlights how absolute numbers can distort intuitive perceptions. A small individual size doesn't prevent a population from reaching a monumental collective scale, as long as individual numbers are high enough to make up for modest body mass. Methods for calibrating between acoustic data and physical catches continue to be refined by research teams around the world. Different species reflect sound in different ways, depending on traits like body size, presence and type of swim bladder, and overall tissue composition. Sophisticated computer models attempt to break down composite acoustic signals into separate estimates for different taxonomic groups present simultaneously in the same sampled region. This calibration work remains ongoing, suggesting that current estimates, while considerably more accurate than earlier ones, may still undergo further adjustments as methodologies continue to be refined. Recognition of this massive biomass repositions the mesopelagic zone as a central, rather than peripheral component of the global ocean biosphere. For decades, scientific and public attention focused predominantly on coastal ecosystems, coral reefs, and surface waters, which are relatively accessible for direct observation. The discovery that this little-known in-between band holds more fish biomass than the entire rest of the ocean combined forces a reassessment of priorities in research, conservation, and potentially future fishing policy, highlighting just how much remains to be understood about a region that, despite its monumental scale, remains invisible to most people. The dominant group within this immense biomass is the lanternfish, or Myctophid family Myctophidae. They're among the most widely distributed, diverse, and populous vertebrates, with some estimates suggesting a total global biomass of 1.8 to 16 gigatons, accounting for as much as 65% of all deep-sea fish biomass. There are more than 250 species in this group, and during the day many stay in lower layers, down to 1,200 meters, swimming up toward surface layers at night to feed on planktonic animals. Typically measuring less than 15 centimeters in length, these small, round-headed lanternfish are pretty unassuming. But what they lack in looks, they make up for in numbers. Every day, in every ocean, billions of these fish swim from the depths to the surface and back, playing a crucial role in the ocean carbon cycle by pulling carbon from the surface and depositing it in the depths. The group's common name comes directly from its most striking feature: light-producing organs, known as photophores, arranged in specific patterns along the body. Each species displays a particular arrangement of these light points, functioning almost like a visual ID code between individuals of the same species under conditions of near-total darkness. This bioluminescence results from chemical reactions involving the protein luciferin and the enzyme luciferase, a process similar to what's seen in land-dwelling fireflies. though it evolved independently in completely different ocean contexts. The light produced serves multiple functions: camouflage through counter-illumination, communication within the species, and possibly attracting mates during specific periods. The considerable taxonomic diversity within the family Myctophidae reflects successful adaptation to practically every ocean on the planet, from tropical waters to polar regions. More than 250 cataloged species occupy slightly distinct ecological niches, varying in preferred depth, specific migratory pattern, and predominant diet. This diversification has allowed the group to exploit virtually every available mesopelagic environment, avoiding excessive direct competition between species through subtle specialization in slightly different resources and spaces. A common evolutionary strategy among successful, geographically widespread taxonomic groups. The ecological role of these fish extends well beyond their own individual survival. Serving as a fundamental link in the ocean food chain, lanternfish are a primary food source for larger predators, including tuna, sharks, large squid, and various diving seabird species. Their massive abundance and relatively predictable availability, thanks to their regular migratory pattern, make them a reliable food resource for multiple upper trophic levels. Without this abundant food base, entire ocean trophic structures would likely collapse or drastically reorganize, demonstrating the disproportionate importance these small fish hold for the ecological stability of much broader marine systems. Their contribution to the global carbon cycle represents another crucial ecological function performed by this group. During nighttime feeding in surface waters, lanternfish consume zooplankton rich in organic carbon derived from photosynthesizing phytoplankton. When they return to the depths during the day, part of that carbon is metabolized, but a significant amount remains incorporated into body tissues or is excreted as sinking fecal matter. This process, multiplied across billions of individuals daily, constitutes a substantial mechanism of vertical carbon transport, contributing to the sequestration of this element at depths where it remains isolated from the atmosphere for considerably long periods. Specific physiological adaptations allow lanternfish to tolerate extreme variations in temperature and pressure during regular daily migrations. Unlike organisms confined to a single depth range, these fish need to adjust their metabolic functions quickly as they move between warm surface waters and cold mesopelagic depths. Adapted enzymes function efficiently across a wide thermal range. Body structures withstand pressure changes without suffering significant damage. This remarkable physiological flexibility represents a sophisticated evolutionary adaptation, enabling efficient exploitation of resources distributed across physically very different environments. Commercial fishing targeted specifically at lanternfish currently remains limited, although growing interest signals possible changes ahead. Their small size and specific body texture make these fish less appealing for direct human consumption compared to traditionally fished species. However, the potential for fish meal production, intended for animal feed or aquaculture, is drawing increasing commercial interest given the impressive population scale recently recognized. This possibility raises important questions about sustainability, considering the fundamental ecological role these fish play in global ocean food chains and biogeochemical cycles. Methods for studying these fish have evolved significantly since the initial discoveries related to the deep scattering layer back in the 1940s. Current researchers combine traditional physical capture, molecular genetic analysis, and continuous acoustic monitoring to better understand the distribution, abundance, and behavior of this extensive taxonomic group. Environmental DNA analysis, a relatively recent technique, allows researchers to detect the presence of specific species through genetic material dispersed in the water, without needing to directly capture the organisms, offering a valuable additional tool for more accurate and less invasive population mapping. Variations in the intensity and pattern of bioluminescence among different lanternfish species continue to be investigated by researchers specializing in animal behavior and sensory physiology. Some species display photophores concentrated predominantly on the ventral side, optimizing camouflage against predators looking up from below. Others show more distributed patterns, possibly related to intraspecific recognition functions during schooling. This diversity of light-based strategies, still incompletely cataloged across all known species, suggests specific evolutionary pressures acting differently on populations occupying slightly distinct ecological niches within the same shared mesopelagic environment. The disproportionate importance of this taxonomic group, relatively unknown to the general public, contrasts sharply with its fundamental ecological role in global ocean functioning. While charismatic species like whales, sharks, and dolphins dominate public and media attention related to marine life, lanternfish remain practically invisible to most people, despite making up the majority share of fish biomass across the entire planet. This gap between real ecological importance and public recognition illustrates a common pattern in the natural sciences, where small, unassuming organisms often underpin ecological structures far larger than their modest appearance would suggest at first glance. While the lanternfish dominates in biomass, another group dominates in sheer number of individuals: the bristlemouth fish of the family Gonostomatidae, especially the genus Cyclothone. Scientists confidently state that this fish outranks every other contender for the title of most common vertebrate on the planet. Ichthyologists estimate the likely number at hundreds of trillions, and possibly quadrillions, or thousands of trillions. The family contains just 8 known genera and 32 species, a relatively small diversity, made up for by extreme abundance. The most prolific genus in the family, Cyclothone, is considered the most abundant vertebrate on Earth, with estimates in the range of quadrillions. Averaging 7.5 centimeters in length, this nearly invisible fish outnumbers the entire planet's birds, mammals, reptiles, and amphibians combined. The common name bristlemouth refers to a distinctive anatomical feature: relatively large jaws equipped with thin, numerous teeth resembling bristles, adapted for catching small prey in an environment where feeding opportunities are sparse and unpredictable. This specialized mouth structure lets the fish maximize its odds of predatory success, even when facing limited, scattered feeding opportunities, a characteristic typical of deep mesopelagic environments where the density of available prey is considerably lower compared to more productive surface waters rich in available photosynthetic nutrients. Differences between species within the genus Cyclothone relate mainly to the preferred depth occupied by each one. Some species concentrate in upper layers of the mesopelagic zone, while others prefer greater depths, extending into the darker, colder bathypelagic regions. This subtle vertical segregation allows multiple species to coexist without excessive direct competition for limited food resources, each exploiting a specific depth range where physical conditions and prey availability favor their particular adaptations. A common strategy among related organisms occupying vertically stratified environments like the deep ocean. Estimating the total population of organisms this numerous and dispersed presents considerable methodological challenges for researchers. Unlike larger species that can be counted individually through direct methods, populations in the range of quadrillions require sophisticated statistical approaches, extrapolating limited sample densities to total ocean volumes. Researchers combine data from trawl catches at specific locations, acoustic calibration, and mathematical models of spatial distribution to generate these monumental estimates. The margin of uncertainty remains considerable, explaining why estimates vary between hundreds of trillions and multiple quadrillions, depending on the specific methodology and assumptions adopted in each scientific study. The ecological role of these tiny fish, despite their small individual size, turns out to be proportionally significant given their extreme abundance. As predators of zooplankton and prey for larger organisms, cyclothone occupies a crucial intermediate position in mesopelagic food webs, connecting microscopic primary producers to predators at higher trophic levels. Its massive, widely distributed presence sustains a considerable population density of predators specialized in catching small prey at this depth range, demonstrating how extreme numerical abundance can offset reduced body size in determining relative ecological importance within a complex ecosystem. Specific sensory adaptations make up for the relative lack of effective vision at depths where light is practically non-existent. Many cyclothone species have reduced eyes compared to other mesopelagic fish, compensating for this visual limitation through highly sensitive lateral line systems, capable of detecting subtle vibrations in the water caused by the movement of nearby prey or approaching predators. This reliance on alternative senses illustrates how different evolutionary lineages, facing similar environmental challenges related to the deep darkness, developed distinct solutions, each optimized for the specific ecological circumstances faced by each particular taxonomic group. The nearly global geographic distribution of these fish reflects exceptional evolutionary success in colonizing mesopelagic environments across the entire planet. Found in practically every ocean, except perhaps the most extreme polar regions, members of the genus Cyclothone show a remarkable ability to adapt to relatively subtle environmental variations between different ocean basins. This cosmopolitan distribution directly contributes to such high total population numbers, since the species isn't restricted to limited geographic regions, but extensively exploits practically the entire available mesopelagic zone on a truly planetary scale. Comparison with the most numerous land vertebrates helps put this extraordinary abundance into context for audiences less familiar with marine biology. Domestic chickens, often cited as the most numerous birds on the planet due to extensive farming for human consumption, total around 25 billion individuals worldwide. That number, while impressive in a land-based context, represents a tiny fraction compared to estimates for cyclothone, which sit in the range of quadrillions. This massive numerical gap highlights how deep ocean environments, largely invisible to everyday human observation, can hold vertebrate populations that exceed any known, documented land-based equivalent by many orders of magnitude. The implications of this extreme abundance for the flow of energy and matter through ocean ecosystems remain an active subject of scientific research. The total biomass represented by trillions of small individuals, even with each one individually contributing modest body mass, adds up to substantial amounts when multiplied by the estimated total population. This collective calculation positions cyclothone as a significant additional component in the overall accounting of mesopelagic biomass, complementing the contributions of lanternfish and other abundant taxonomic groups present simultaneously in this same in-between depth range shared by multiple species adapted to similar conditions. The gradual discovery and recognition of this fish's extraordinary abundance, despite being practically unknown to the general public, illustrates a recurring pattern in modern biological oceanography. Small, unassuming, historically understudied organisms often turn out to be numerically dominant components of entire ecosystems, once adequate sampling and quantification technologies become available to researchers. This ongoing upward revision of population estimates, as scientific methods improve, suggests that our current understanding of the abundance of life in the mesopelagic zone, while already impressive, possibly still underestimates the true biological scale present in this vast in-between region of the planet's oceans. The enormous concentration of prey in the mesopelagic zone doesn't go unnoticed by the large predators that normally live in sunlit surface waters. These middle depths are well within reach of many top predators, including those that live predominantly in the epipelagic zone and make feeding forays into greater depths. This includes many sharks. Recent studies assessing the importance of mesopelagic prey for 3 top teleos predators in the northwest Atlantic confirm this zone's central role in the diet of commercially valuable, highly migratory predators. Tuna, swordfish, and sharks regularly dive beyond their usual surface habitat, specifically to feed on the concentration of lanternfish and bristlemouth that rises and falls daily. A food supply so predictable and abundant that it shapes the migratory behavior of entire species of large ocean predators. Picture a predator built for hunting under abundant light, deciding to dive hundreds of meters toward the cold dimness just to access concentrated food. This behavior requires tolerance for sharp temperature swings, reduced sensory efficiency, and resistance to greater pressures than at the surface. Species capable of these forays developed, over generations, body traits that make repeated dives possible without compromising vital functions, balancing the energy cost of the trip against the caloric payoff of catching abundant prey in this in-between layer of the ocean. Large tuna keep their muscle temperature elevated even in cold, deep water, an uncommon trait among fish. A specialized circulatory system, called a rete mirabile, retains the metabolic heat produced by muscle activity, acting as a heat exchanger that reduces heat loss to the surrounding water. This adaptation makes repeated forays into mesopelagic depths possible without compromising the vigorous swimming needed for both hunting and escaping threats, allowing these predators to regularly exploit an environment that's physiologically hostile to their usual warm water metabolism. Swordfish have elongated bills and eyes adapted for low light, representing another group specialized in exploiting mesopelagic resources. Special eye tissues warm the retina and brain, improving visual processing speed even under the reduced temperatures of the depths. This adaptation allows for quick reaction to moving prey, even under fairly reduced lighting compared to the surface environment. Processing visual information quickly in the deep cold offers a significant competitive advantage for catching agile prey, such as lanternfish, during regular feeding forays beyond these predators' usual habitat. The diversity of sharks that dive down into the mesopelagic zone is surprising given the taxonomic breadth involved. From medium-sized species to some of the largest ocean predators, multiple lineages have independently evolved the ability to exploit this abundant food source. Electroreceptive sensors, capable of detecting weak electric fields generated by the muscles of moving prey, make up for visual limitations in dark environments. This additional sensory ability, combined with a keen sense of smell, allows them to locate concentrations of prey even when conventional vision loses nearly all effectiveness in the dimness characteristic of this in-between depth band. The timing patterns of these predatory forays often coincide with the very migratory rhythm of their target prey. Predators adjust their dive schedules to maximize overlap with the times when mesopelagic fish concentrate at specific depths, optimizing capture efficiency per unit of effort spent. Telemetry tracking studies, using electronic tags attached to temporarily captured individuals, have revealed these patterns with growing precision over the past few decades. Data show dives synchronized with times of deepening dusk or dawn, moments when prey layers are in transition between surface and mesopelagic depths. The high commercial value of tuna, swordfish, and various shark species gives direct economic relevance to this predatory relationship with the mesopelagic zone. The global fishing industry depends heavily on these species, whose availability and distribution are closely tied to the abundance of mesopelagic prey that sustains their regular diet. Changes in this in-between layers biomass, whether from direct commercial exploitation or climate-driven shifts affecting ocean productivity, could directly impact populations of these valuable predators, creating a meaningful economic connection between a little-known ocean layer and fishing industries operating on an international scale. Satellite tracking technologies, combined with depth sensors attached to tagged individuals, have made it possible to document the frequency and depth of these feeding forays in unprecedented detail. Data accumulated over the years reveals that some individuals make dozens of mesopelagic dives each month, each one lasting from minutes to a few hours, interspersed with extended periods in warmer surface waters. This pattern suggests a calculated energy strategy. Predators briefly tolerate the physiologically challenging conditions of the depths, quickly returning to the thermal comfort zone of the surface once feeding is successfully completed. The ecological implications of this connection between surface predators and mesopelagic biomass extend to practically the entire ocean trophic structure. These predators function as a direct link between energy captured in the depths and even higher trophic levels, including seabirds and marine mammals that in turn prey on tuna and swordfish at certain life stages. This chain of energy transfer, originating in surface photosynthetic primary production and crossing multiple vertical layers of the ocean, illustrates the deep interconnection between seemingly distant and disconnected regions of the marine environment, all united by the daily movement of biomass between the mesopelagic zone and sunlit surface waters. Growing understanding of these predatory forays underscores how much the mesopelagic zone, despite being barely visible and rarely mentioned outside specialized scientific circles, directly sustains economically important species that are ecologically central to healthy oceans. Responsible fisheries management needs to account for this indirect dependence, recognizing that protecting tuna or shark populations eventually also requires attention to the integrity of the mesopelagic layer these predators regularly depend on. This network of vertical connections, linking depth and surface through repeated daily dives, stands as a striking example of how the ocean functions as an integrated system, rather than as isolated compartments with no relationship to one another. The daily movement of billions of organisms between the surface and the mesopelagic zone has a consequence that goes well beyond marine biology. It regulates part of the planet's climate. As these organisms rise at night to feed and return to deeper waters during the day, they excrete waste and many die, effectively moving carbon downward, often sequestering it on the deep ocean floor, where it can remain for centuries. This process, known as the biological carbon pump, plays a vital role in mitigating the effects of carbon dioxide in the atmosphere. Vertical migration provides a critical energy link between surface waters and the deep ocean. Marine animals that migrate vertically transport carbon from the surface to the deep ocean, an important process in regulating global climate. Trillions of small fish swimming up and down every single night collectively function as one of the largest natural carbon capture mechanisms on Earth. Picture the carbon cycle as a continuous transport system, where this element circulates between the atmosphere, the ocean surface, and the deep sea through interconnected physical, chemical, and biological processes. Phytoplankton, microscopic and photosynthetic, capture atmospheric carbon dioxide dissolved in surface water, converting it into organic compounds through photosynthesis. This incorporated carbon enters the food chain when zooplankton and small fish consume phytoplankton. Vertical migration then actively transports this captured carbon down to depths, where it remains isolated from the atmosphere for considerably longer periods, completing a fundamental step in the biogeochemical cycle that regulates atmospheric concentrations of this greenhouse gas. Specific transport mechanisms include several distinct pathways, each contributing to the downward movement of carbon. Respiration by organisms that feed at the surface and descend to mesopelagic depths releases carbon dioxide directly into these deeper layers, effectively removing it from the atmosphere near the surface. Fecal matter produced by these animals, rich in undigested carbon, continuously sinks, forming a significant part of what scientists call marine snow. Carcasses of organisms that die during or after migration also contribute, sinking and depositing carbon stored in body tissues at depths where decomposition happens much more slowly than near the sunlit surface. Marine snow, a descriptive term for organic particles that continuously sink through the water column, includes not just fecal matter, but also dead phytoplankton cells, remains of various organisms, and mucus aggregates produced by certain species. These particles, visually resembling snowflakes slowly drifting down through a dark aquatic environment, carry organized carbon from the productive surface down to depths, where most of it is eventually consumed by detritivorous organisms or chemically breaks down. The fraction that escapes this intermediate recycling and reaches the true ocean floor can remain sequestered for extended geological periods, effectively removing that carbon from active atmospheric circulation for centuries or millennia. Precisely quantifying DEAL vertical migration's contribution to total ocean carbon sequestration presents a considerable scientific challenge, given the complexity of measuring flows on such a vast, globally distributed scale. Researchers use a combination of direct measurements through sediment traps positioned at different depths and computer models that integrate data on migratory biomass, metabolic rates, and behavioral patterns. Current estimates suggest that this specific biological mechanism may account for a substantial fraction of the total carbon exported annually from surface waters to ocean depths, although the exact figure remains subject to ongoing refinement as methodologies improve. Comparing the biological carbon pump to other natural carbon capture mechanisms helps put its relative importance into context. Land forests, often highlighted in discussions about climate mitigation, capture carbon through photosynthesis and storage in plant biomass and soil. Oceans, through combined physical and biological mechanisms, including DEAL vertical migration, capture a comparable or potentially greater amount of atmospheric carbon over annual periods. The key difference lies in the storage timescale. Carbon sequestered in ocean depths can remain isolated for much longer periods than carbon temporarily stored in land vegetation, which is subject to relatively faster decomposition. The sensitivity of this process to environmental change worries scientists studying the interactions between oceans and global climate. Changes in surface temperature, progressive ocean acidification, and shifts in phytoplankton primary productivity, all potentially influenced by ongoing global warming, could significantly affect the efficiency of this biological pump. If populations of migratory organisms decline due to environmental stress or excessive commercial fishing, the ocean's ability to sequester carbon through this specific mechanism could be compromised, creating a potential feedback loop where climate change weakens one of the very natural processes that helps mitigate that same change. Recent research using more sophisticated acoustic tracking technologies has allowed for refined estimates of the specific contribution different taxonomic groups make to this carbon transport. Lanternfish and bristlemouths, given their extraordinary collective biomass, likely account for a substantial share of this flow, though migratory zooplankton also contributes significantly through similar mechanisms. Differentiating the specific contributions of each group remains an active area of research, with direct implications for understanding how changes in specific populations, whether from commercial exploitation or environmental shifts, could affect the total carbon sequestration capacity of this in-between ocean region. Implications for climate and marine conservation policy are becoming increasingly clear as scientific understanding of this process deepens. recognizing the mesopelagic zone as an active, significant component of the planetary climate system, rather than just an isolated biological habitat, strengthens the case for protecting this region from unregulated commercial exploitation. International organizations dedicated to ocean policy are beginning to factor considerations about the biological carbon pump into discussions on sustainable management of marine resources, recognizing that this ocean layer's value considerably exceeds its potential as a direct food source or industrial raw material. Growing understanding of this connection between everyday animal behavior and planetary climate regulation illustrates how seemingly local biological processes can have truly global consequences when multiplied across the appropriate scale. Trillions of small organisms, repeating the same vertical movement night after night, collectively influence how much carbon stays in the atmosphere versus how much gets sequestered in the ocean depths for centuries. This natural mechanism, operating silently on a planetary scale long before any formal scientific understanding of its existence, continues working without interruption, connecting the feeding behavior of tiny fish to climate processes that directly affect all life on the Earth's surface, including the human species, which has only recently begun to properly recognize and appreciate this fundamental connection. Faced with a biomass estimated at billions of tons, the global fishing industry has repeatedly tried, since the 1970s, to make commercial harvesting of mesopelagic fish viable. Two major large-scale research projects were launched in September 2019 to investigate the feasibility of these fisheries, funded by the European Union with a budget of more than 13 million euros. Despite this interest, commercial exploitation remains extremely limited. There are no commercial fisheries targeting the hundreds of different lanternfish species, even though they're estimated to be among the most abundant fish in the mesopelagic zone, with an estimated biomass of around 600 million metric tons. The difficulty isn't finding the fish, it's processing it, small, fragile, oil-rich bodies that deteriorate quickly and don't fit traditional industrial fishing methods. Picture a modern fishing fleet, equipped with technology capable of precisely locating schools through advanced sonar, finally positioning nets over a biological layer containing billions of tons of fish. Despite this technical ability to locate the fish, catching them in an economically viable way turns out to be a completely different problem. Mesopelagic fish, adapted to the pressures and temperatures of the depths, often suffer severe physical damage during the rapid ascent to the surface, a process that ruptures delicate tissues not built to withstand sudden pressure changes. This structural damage drastically compromises the quality of the final product, making subsequent processing considerably harder compared to fish caught in conventional surface waters. The body composition of these organisms creates additional challenges for traditional fishing methods, historically developed for larger, sturdier species. Many mesopelagic fish contain a high proportion of oil in their tissues, an adaptation that helps with neutral buoyancy in an environment of varying pressure. This high-fat content, while biologically advantageous for the organism itself, speeds up spoilage after capture, requiring nearly immediate processing to avoid significant quality loss. Conventional fishing infrastructure, designed to transport and process catch over longer time frames, doesn't easily accommodate this near-instantaneous processing requirement characteristic of these particular species. The small individual size of lanternfish and bristlemouths represents another significant obstacle to the economic viability of large-scale capture. Conventional nets, sized for larger commercial species, often can efficiently retain organisms this small without also catching excessive amounts of other unwanted species, a phenomenon known as bycatch. Subsequently sorting target species from bycatch requires additional processing, raising operational costs. On top of that, the volume needed to make the operation economically viable, given the tiny size of each individual, would require a capture scale that raises serious questions about long-term ecological sustainability. European projects funded starting in 2019 specifically sought to develop technologies and methods capable of overcoming these historical technical limitations. Researchers investigated alternative capture methods, onboard processing aboard specialized vessels, and potential commercial applications for the final product, including fish meal, oil for supplements, and possible pharmaceutical uses. Results from these projects indicated that, while technically possible to overcome some capture and processing challenges, full economic viability remains questionable without substantial subsidies, given the high costs associated with the specialized operations needed to properly handle the unique characteristics of these organisms. Parallel ecological concerns add to the technical and economic difficulties involved in commercially exploiting this biomass. Scientists repeatedly warn that the crucial ecological role these fish play, both in the food chain and in carbon transport via the biological pump, remains insufficiently understood to ensure that large-scale extraction won't cause unwanted and potentially irreversible consequences. Unlike traditional fisheries, where decades of historical data inform sustainable management practices, mesopelagic exploitation would initially lack that accumulated knowledge base, considerably raising the risk of management mistakes with broad ecological impacts that would be difficult to reverse later. Earlier attempts at commercial exploitation, predating the more recent European projects, often failed for similar reasons related to high operational costs compared to the relatively low market value of products derived from these species. Unlike fish traditionally consumed directly by humans, which command higher market prices, products derived from mesopelagic fish are mainly intended for lower value markets, such as animal feed or industrial oil. This combination of high capture and processing costs with a relatively low final product value creates a narrow profit margin, making investment in this proposed industry considerably risky for interested commercial operators. Emerging technologies continue to be developed in an attempt to work around these historical limitations. Gentler capture systems, specifically designed to minimize physical damage during collection, represent one active line of research. Immediate onboard processing, using compact equipment capable of converting catch into stable products even before returning to port, offers another potential approach to getting around rapid spoilage problems. Despite these ongoing technological innovation efforts, a truly viable commercial scale remains elusive, suggesting that this massive biomass will likely remain largely commercially unexploited in the near future, at least using currently available technological approaches. Scientific debate over whether this biomass should be commercially exploited, even if technically feasible, remains active among researchers and marine policymakers. Arguments in favor highlight the potential to meet growing demand for animal protein and industrial oils without putting further pressure on traditional fish stocks already overexploited in many regions. Arguments against emphasize the substantial ecological risks tied to removing massive quantities of organisms whose role in global biogeochemical cycles has only recently begun to be fully recognized. This tension between economic potential and ecological caution will likely continue to shape regulatory decisions related to this biomass in the years ahead. The current situation reflects a notable paradox. One of the largest concentrations of animal biomass on the planet remains, at the same time, virtually untouched by the commercial fishing industry and scientifically recognized as a potentially valuable resource. This combination of extreme abundance and practical inaccessibility illustrates how significant scientific discoveries don't always translate directly into viable economic exploitation. especially when the fundamental biological characteristics of the organisms involved directly clash with industrial methods developed for completely different contexts, leaving this vast biomass essentially preserved not by any deliberate conservation decision, but by technical and economic limitations not yet overcome. The same phantom bottom that confused U.S. Navy sonar in 1942 turned out, decades later, to be the largest concentration of vertebrates on the planet. The mesopelagic zone is the cold, dimly lit ocean layer found between 200 and 1,000 meters below the surface, which scientists say plays key roles in the marine food web and the carbon cycle. The lanternfish rises every night and descends every day, carrying carbon down into the depths. The bristlemouth, possibly the most numerous vertebrate on Earth, makes the same trip in numbers that exceed any human population estimate. Tuna, sharks, and swordfish dive beyond their natural habitat just to reach this abundance. And the sonar that in 1942 registered a floor that didn't exist had detected, without anyone knowing it, the largest synchronized biomass migration that Earth produces every single day, in a space that covers 60% of the planet's surface and remains, for the most part, unexplored. Picture sonar operators of that era, watching a persistent, confusing echo on their screens, with no conceptual tool available to correctly interpret what they were actually recording. Decades of subsequent research, involving oceanographic expeditions, specialized nets, and increasingly sophisticated acoustic technologies, were needed to turn that initial confusion into solid scientific understanding. This path, from accidental discovery during military operations to full recognition of this layer's ecological and climatic importance, illustrates how scientific knowledge often advances through incremental steps, each discovery building on foundations laid by earlier investigations, often driven by purposes completely different from those that eventually turn out to matter. Recognition of the biological scale present in this in-between band, later revised upward through modern acoustic technologies, completely redefined how scientists understand the distribution of life in the oceans. Estimates that place mesopelagic biomass between 10 and 15 billion metric tons substantially exceed all of humanity's annual fishing catch combined, revealing a biological reservoir of a magnitude few imagined existed before systematic investigations confirmed this reality. Lanternfish, accounting for the majority share of this biomass, and bristlemouths, possibly outnumbering all other land and marine vertebrates combined in absolute number of individuals, exemplify how individually modest organisms can collectively constitute a truly dominant biological presence on a planetary scale. The connection between these organisms' daily migratory behavior and global climate regulation represents perhaps the most surprising aspect of this still unfolding scientific story. Billions of small fish, repeating the same pattern of nighttime rise and daytime descent for countless generations, collectively transport substantial amounts of carbon from surface waters down to depths where it stays isolated from the atmosphere for considerably long periods. This natural mechanism, operating silently long before any formal scientific understanding of its existence, now stands as a recognized, relevant component within climate models that seek to understand and predict the future behavior of Earth's climate system in the face of rising atmospheric greenhouse gas concentrations. Larger ocean predators, including tuna, swordfish, and various shark species, developed over generations specific physiological adaptations that make regular forays beyond their usual surface habitat possible, specifically to exploit this predictable, abundant food concentration. This dependence, documented through detailed studies on diet and migratory behavior, directly links the health of commercially valuable populations to the ongoing integrity of the mesopelagic zone. Any significant change to this in-between layer, whether through direct commercial exploitation or broader environmental shifts, would likely ripple through multiple trophic levels, affecting even species that are economically important to international fishing industries. Attempts at commercial exploitation targeting this massive biomass, despite decades of interest and research investment, continue to run into significant technical and economic obstacles related to the specific biological characteristics of the organisms involved. Small, oil-rich bodies prone to rapid spoilage don't easily fit industrial fishing methods developed for completely different contexts. This practical difficulty, combined with legitimate scientific concerns about the potential ecological consequences of large-scale extraction, keeps this biomass essentially preserved, not by any deliberate conservation decision, but by technical and economic limitations that persist despite considerable investment in research and technological development aimed specifically at overcoming these challenges. The global distribution of this zone, covering roughly 60% of the planet's surface and 20% of total ocean volume, positions it as one of the largest continuous environments on the planet, exceeding any known land biome in extent. This monumental scale, combined with relatively recent recognition of its ecological and climatic importance, contrasts sharply with the still incomplete level of scientific exploration dedicated specifically to this region. Much of the ecological processes, behavioral patterns, and interactions between species present in this layer remain only partially understood, suggesting that future investigations will likely continue revealing additional aspects of how this vast in-between region of the world's oceans functions. The scientific methods used to study this zone have evolved considerably since the confusing sonar readings of the 1940s, now incorporating sophisticated acoustic technologies, molecular genetic analysis, satellite telemetry applied to migratory predators, and complex computer models capable of integrating data from multiple sources simultaneously. This methodological evolution has allowed for substantial refinements in population estimates, understanding of migratory patterns, and quantification of carbon flows associated with these organisms' behavior. Each technological advance in this direction tends to reveal additional complexity, suggesting that current understanding, while considerably more robust than decades ago, still represents only a fraction of the knowledge potentially available about this extensive ocean region. The implications of this growing understanding extend beyond purely scientific interest, reaching into discussions about marine conservation policy, sustainable fisheries management, and climate mitigation strategies on a global scale. Recognizing the mesopelagic zone as an active, significant component of both the ocean food web and the biogeochemical carbon cycle strengthens the case for protecting this region from unregulated commercial exploitation, while also acknowledging its legitimate potential economic value should sustainable exploitation methods eventually be developed. This balance between protection and potential use will likely continue shaping international policy related to this region in the years ahead. The trajectory of this scientific discovery, which began as technological confusion during military operations and has culminated in recognition of its fundamental role in planetary climate and ecological function, remains a work in progress as new investigations reveal additional aspects of this vast, intricate region. The sonar operators of 1942, watching that persistent, unexplainable echo on their screens, could never have anticipated that they were detecting, with no understanding at the time, what would later be recognized as a fundamental natural mechanism connecting the feeding behavior of trillions of small organisms to processes that directly shape climate and life on a truly planetary scale, showing how seemingly accidental discoveries often pave the way for scientific understanding that few could have anticipated at the outset.